Okay, thank you. Good morning, everyone. Thanks to the Pages Human Traces Working Group organizers for inviting me to give this talk today. I attended yesterday America session and have to say there were an amazing lineup of speakers and topics. Today, I would like to present an overview of my research focused on understanding anthropogenic impacts over time using various statistical techniques and combining disparate data sets, spatially and temporally, to basically understand when, where, and by how much aquatic ecosystems have changed. So I basically be focusing on some coastal wetlands in the Mediterranean Sea, but also my latest research in tropical South America. So that's not totally Europe-centric based research I'll be talking about today. Okay, so here, yeah. So no big news here. So the world is changing at fast rates and magnitudes, and everyone knows it. So this is the famous hockey stick plot of Willa Stephan on rapid change in the Anthropocene. Just yesterday, Emily presented or showed as the blue acceleration, which is the analogous of this rapid change acceleration in the marine realm, but the same conclusion can be drawn. There exists a wide array of steep changes in drivers in terrestrial, freshwater, marine, and more importantly, in socio-ecological systems. So we have global problems here that requires, of course, global approaches. So over my research, I've been working with aquatic proxies, basically diatoms and benthic foraminifera. So these two organisms are mostly at the base of many aquatic food webs, so they may act as the first responders to changes in the aquatic conditions. Obviously, diatoms, because they are algae, so they are sensitive to changes in nutrient salinity and other conditions. They are found in every single water body on Earth, from freshwater to transitional waters to marine ecosystems. And on the other side, benthic foraminifera, because they are protozoes, so they are not so sensitive to nutrients, they might be indicative of other anthropogenic impacts such as eutrophication, organic matter deposition, and perhaps more importantly, sediment type and sediment provenance, because they are mostly restricted to the coastal zone, and they can be really, really good indicators in these transitional waters. No big news again, both are useful paleoecological proxies. They can be preserved in the sediments for long periods of time due to the siliceous and calcium carbonate cell walls. Aquatic ecologists, as myself, tend to have a strong, let's say, wishing in first putting together what we call training sets to better understand what are the autoecology of the species for informing better the long-term evolution and anthropogenic impacts in the record. This is what exactly we did in the Ebro Delta in Spain. This is one of the largest deltas in the Western Mediterranean. It acts as a natural laboratory, as I like to call it, because it's highly sensitive to sea level rise, sediment loss by damming in the last portion of the river. But also, as you can see, 80% of the total delta surface, these tiny green polygons are rice fields. So as you can imagine, all the agricultural runoff waters goes directly to natural habitats, and the hydrological regime is highly impacted and altered by these human activities. We put together a training data set combining diatoms and foraminifera to try to tease apart which are the main gradients that structure species abundance and composition. Also, many aquatic ecologists tend to build regression models to reconstruct single estimates of variables that are indicative of any type of or some types of human activities and natural factors as well. And we built a regression model transfer function for conductivity to better estimate changes in conductivity as freshwater inputs are the main factor explaining diatom distribution here in the delta. But one of the main issues that we encounter is that in these dynamic ecosystems, single estimates might not well indicate the habitat structure, and hence we couple this regression model with more qualitative approaches, indicator species analysis, to tease apart different types, habitat types for the same level of conductivity to get a much more nuanced picture of the history in the delta. Exactly the same reasoning can be applied to benthic foraminifera, in this case using water depth transfer function. For the delta plate gradient, very shallow habitats, water depth is not a good, let's say, primary driver of anthropogenic impacts, but instead benthic foraminifera arranged pretty well following an organic matter gradient. And this allowed us to get a much more nuanced reconstruction of the delta over the last 3,000 years, following mostly changes in the river mouth. However, one of the key questions when using trying to quantify ecological data on indicator taxa is determining the geographical extent of sites and how well the ecological characteristics of taxa are quantified, right? A serious challenge when we try to expand or extrapolate this autoecology of the species in other areas in thinking about large scale is the correlation between environment, climate, and space. So we face with multidimensional data, multi-species composition. So a natural question would be, is a species not found in a particular habitat only because it was not dispersed there, or the environmental conditions weren't suitable for the species to grow? And these species have been driving me crazy over the last years, and drove, I would say, my most current research. This is when we cross the pond and go to tropical South America, and we built this tropical South America diatom database here in this plot, showing you the distribution of the sites, mostly lakes, rivers, and streams associated with environmental data from the lakes, but also this more regional context to get more, let's say, characteristics of climate and also aquatic connectivity. This database is available for the community in GitHub, and we are working with the Neotoma database stewards to make it also available in there. We built a couple of Shiny apps for visualization, so to see where the species are present and which are the main environmental variables and geoclimatic factors for paleoecological reconstructions. So I'm going to go now for a couple of examples to use how we can use this database, thinking about more in terms of this working group building together and synthesizing regional studies. So we wanted to test to what extent planktonic diatoms, because they are highly sensitive to physical structure changes in lakes, like thermal structure due to warming and also nutrient inputs, may be indicative of anthropogenic impact. So different planktonic species showed quite wide ranges in thresholds for different variables like water temperature and mostly conductivity. Another important thing, if you remember the Venn diagram I showed you before, using variance tuning, we could predict to what extent broad scale factors may even outperform the variance explained by water chemistry in lakes. So we need to take into account factors associated with dispersal when working with aquatic indicators to be considered for quantifying ecological anthropogenic impacts. Another question that we wanted to ask using this database is to what extent lakes and by inference, so diatoms and by inference lakes were resilient or sensitive to stressors associated with long-term occupation in the basin. And we used an index called local contribution of beta diversity to assess what extent different predictors explain continental scale beta diversity of diatoms. The take-home message here is that diatom communities in historical impact basins, dry and alkalophilous lakes, are the ones who contributed more to this continental scale beta diversity. We don't know exactly when, but the results show that lakes located in central Chile and the Peruvian Altiplano have significant value. So intensity of past human impacts are higher in that portion of the latitudinal gradient. So to dig into more detail about when and by how much humans started to impact or alter the lake ecosystems, we wanted to expand this database and incorporate different components of the human dimensions of environmental change. And for that, we started to collate other data sets indicative of the archaeology and mostly the vegetation change, charcoal, and also several geospatial data sets that are available in the literature, like line hues and human densities estimates for the Holocene, to get a more broader picture and more nuanced explanation of the human changes. That was a huge task because although from the theoretical point of view, the community called for these interdisciplinary approaches, is really difficult and challenging to link different time series of proxies of past change, especially different taxonomy and also spatial and temporal mismatches. But I would like to show you a couple of examples of this newly constructed database more interdisciplinary point of view. So we analyzed 4 high-resolution sediment cores in Ecuador. These 4 lakes are limnologically and spatially distinct in nature, and we wanted to look at coherent signatures of human impact in the Anthropocene and how those differ from the previous time. So let's say over the long term or the pre-Anthropocene, for the sake of today's workshop and this working group. So we analyzed trends of the diversity time series and correlate with drivers of climate, delta O18, and human factors archaeologically recorded radiocarbon time series, and also cropland area and human density from the HIDE data set. So the take-home message here is that anthropogenic impacts over the last few centuries, we observed that they accelerated and even synchronized abrupt rates of diatom change in the less remote lakes, which are Yaviucu and Yaguarcocha. But those shifts occurred after long periods of ecological stability. So that was quite intriguing for us. And we interpreted these lakes as may exhibit resilience to a variety of stressors. So we wanted to ask then, why are some systems more prone to be synchronized by human activities, while others are more resilient, or even they don't respond to well-known either climatic changes and human occupations in the basin. So for the last part of my talk, I hope I'll have time to explain a little bit. I would like to talk about ecological resilience and how we can develop this or incorporate this framework into the Anthropocene and pre-Anthropocene human impacts in ecosystems. Here I'm showing you the famous ball and cup analogy. So on the left, we have a high resilient state, which is depicted by a deep cup, and the ecosystem is the ball. So any time an ecosystem get disturbed, it returns rapidly to an equilibrium state after perturbation. On the right, though, a low resilient state is characterized by a much shallower basin of attraction, and therefore any perturbation that the ecosystem phase may move it to an alternative stable state, right? And it might take more time to return to the equilibrium. This whole concept of ecological resilience has been developed using monitoring data. So people have been adding nutrients in manipulated lakes and model when the change is observed. But I believe the same reasoning can be applied, of course, taking into account the multiple idiosyncrasies of paleoecological time series to the proxy realm. And then we may have a full catalog, not only 2, but perhaps multiple basins of attractions, right? And we might have a catalog of different drivers that can be indicative of different human occupations, acceleration, and inceptions in the ecosystems. So we tested that idea of regime shift and abrupt changes, modeling rate of change. So rate of change are supposed to be an indicator of how resilient or loss of resilient states and to what extent the ecosystems are approaching to an impending change, using a recent time series analysis of nonlinear rate of change using generalizative models. So very rapidly here, what we observe is that so you have to see here in the column of richness, and do not take too much to count the turnover component, but many lakes, so these are the same 4 lakes in Ecuador, many lakes shows ups and downs, so they are not showing abrupt, so rapid changes in diatom species composition, but a Lake Yaobiuku at the bottom, it becomes, for the most part of the pre-Anthropocene over the same regime, but just entering in Anthropocene the last 500 years. In this case, it shows an abrupt change, or it might enter into an alternative stable state. Of course, this is a cool method, but we don't know which are the proximate drivers of change. So we can combine other types of multivariate ordination techniques like PCA and add different layers of information. So here I'm showing you a principal component analysis of the diatom training set in tropical South America. On the top of that, we have the fossil core trajectory. So how over time species and communities are changing. And on the top of that, we are adding isolines of total phosphorus. So anytime the core trajectory cross one of these isolines, we might expect the species reacting to some sort of threshold in this environmental gradient. If we link this with the analogy of a ball and cap and trying to quantify resilience, the 2 lakes that are supposed to be more stable over time, Piñana and Fondococha, there seems to be within their basin of attraction because the cloud points are really close to each other. In other cases, for instance, Yaguarcocha might even return to an initial state following eutrophication, and Yabiuco seems to be following to another basin of attraction because it crossed several times the total phosphorus gradients. Okay, so the last thing I would like to give you today about how we quantify resilience to stress the importance of change not in mean but also in variance and as another indicator of loss of resilience in paleoecological records, and to get a continuous metric of change over time is variance. So we can model variance using GAMs as well. So different components of the ecosystems, diatoms, pollen, and agropastoral indicators coming from human activities, mostly charcoal, sporoprimella, and also ZMAs, pollen grain, when they appeared first in the record. It shows ups and downs, but here I would like to show you that for the last 500 years in Lake Yabiuco, we observe a quite steep decrease in variance following European contact. So lakes are quite responding to human activities or inception or even acceleration in this portion of the lake. Okay, so the conclusions. I don't know if I'm running out of time. Very briefly, I think you can read here what I wanted to summarize, but I would like to give you a couple of announcements, and I think it's quite timely for today's workshop and the beginning of the Human Traces Working Group. This is very timely because many community-curated databases are becoming available. So global scale projects like this help us integrate disparate knowledge and about different dimensions of human change. And from Pages, we are also working on a joint early career workshop, the past social environmental systems. Unfortunately, we had to postpone 2 times the in-person workshop in Chile, but we have been working online in several punctuate events. And just recently, yesterday, we were granted with a small grant by INCWA about building a synthesis database of past human environmental systems in the global south. So I think we are multiple joint interests here, and I will be very happy to discuss with you all later. And with that, I will take any questions that you might have. Thank you so much for your attention.
Yeah, thank you, Javier. That's fantastic. Really interesting. We have a very short amount of time for a question. Otherwise, people could perhaps type their questions into the chat and we can address them a little bit later.